Hello, everyone. Welcome to the Introduction to Computational Neuroanatomy at the DIPY Workshop 2021. This is the first part of this introduction, and I will focus on the connection between neuroanatomy and diffusion tractography. In part 2, Francois Riel will focus more on the basics of tractography. So we know that DIPY is a tool for diffusion MRI analysis to study the white matter in the brain. But why do we care so much about the white matter in the first place? A major reason is that the white matter is composed of connections between neurons. We know that neurons are the basic computing units in the brain, and they communicate with each other through axons. Most axons are wrapped in myelin, which is the material that make them look white. So studying the white matter is to study the neural connections, and how neurons are connected is essential for their computations. Here is an example of a brain network underlying obsessive compulsive disorder. Indeed, almost all brain diseases involve abnormality in more than one region. This indicates that to fully understand the dysfunction, we have to take into account not only the regions, but also their connections. On the right-hand side is another example from a study on mood disorder. These are the FA maps showing abnormality in patients, and these abnormalities are colored in red and blue. The low FA values indicate impairment in the white matter integrity. You can see that the impairment is localized precisely in these confined regions in the white matter. This means that if we want to develop treatment that targets these abnormalities, we need the precise information of brain connectivity in those regions. Now let's follow up the OCD example and look at an effective treatment procedure called deep brain stimulation. This procedure uses implanted electrodes to deliver currents to particular sites in the brain. The sites that are responsive to the stimulation have been found in both gray and white matter. One particularly responsive region is the so-called anterior limb of an internal capsule. This is a small white matter volume buried in the striatum. The striatum is part of basal ganglia, which is a subcortical structure. You can see the shape of striatum in the right figure, which is colored in purple and blue. The gap in between is the internal capsule. You can see the electrodes are targeting the anterior part of the internal capsule. If the physician is planning a surgery to implant the electrodes, it is very important to accurately locate not only the internal capsule, but precisely the part of it that connects the regions related to the OCD symptoms. So how do we figure out the detailed connections? This is the exact place neuroanatomy can come to help. Just a little bit history of the neuroanatomy research in humans. It used to be a postmodern science. The most widely used method is named after its inventor, Klinger. The procedure is to first freeze and thaw the brain tissue, and then systematically peel away white matter fibers to expose the target tracts for investigation. In this way, one can examine the trajectories of major fiber bundles in the whole brain. Klinger's method had dominated the field for decades until the 1990s, when magnetic resonance imaging and modern microscopy started to take off. Before we delve into diffusion tractography, here are two microscopy techniques that's worth mentioning. One is polarized light imaging. The other is optical coherence tomography. Both of these techniques use light reflection or refraction properties to delineate the fibers at a micrometer resolution. Like Klinger's method, these are relying on postmortem tissues as well. Around similar times in the 90s, the diffusion imaging technique was starting to be developed as well. Its resolution is higher than Klinger's method and lower than the microscopy methods. But most importantly, this is an in vivo method. Until today, diffusionmri is still the only non-invasive method to map fiber pathways in a living brain. Here is briefly how it works. The diffusion-weighted signal reflects the direction of water diffusion in the brain. In the axons, water diffuses most easily along the long axis, thus generates strongly anisotropic diffusion signal. Based on this signal, one can model the most plausible orientation of axons in different locations of the brain, and put these pieces together, one can infer the trajectories of axons. So have we achieved the goal of mapping every fiber pathway in the human brain with the excellent technology? To gain some sense of the big picture, here is a review that came out last year, which did a survey of 3 authoritative neuroanatomy textbooks. The survey provided the percentage of studies cited to support each pathway mentioned in the books. Somewhat surprisingly, the majority of our current knowledge comes from animal studies. That is, we infer the pathways in humans from observations in animals by assuming cross-species homology. This means a large part of the human brain connectivity is still unknown. To quote the authors of the review, one essential aspect of the human central nervous system structure is virtually unknown, precisely where a specific connection originates and terminates in the brain. What is stopping us from understanding the human brain directly? A limitation of diffusion tractography is that it's an indirect way to measure anatomical connections. The same diffusivity pattern can be generated by different configurations of axons. Thus, there is no unique solution to the inference problem. For example, in the schematic drawing shown on the right here, the water diffusivity points to 2 dominant directions. If an estimated fiber pathway reaches this crossing point, how do we know whether it should continue straight, turn right, or turn left? Without additional information about the underlying anatomy, there is no way to decide solely based on the diffusion model. However, there is only one anatomical ground truth. This means that diffusion tractography is prone to errors in the regions like this with crossing fibers. Here is what animal studies can help. To solve the problem, we need some prior knowledge of what the ground truth is most likely to be. And most of this knowledge comes from humans' closest relatives, the non-human primates. When we look at the history of neuroanatomy, there is a parallel branch dedicated to animal research. The very early methods relied on staining techniques, and they are applicable to both human and non-human brains. These techniques are not sensitive enough to distinguish microstructures. In the 1950s, a type of selective staining emerged. It worked by introducing degeneration to specific groups of neurons in living animals and staining the degenerated pathway in the postmortem brain. This method is so much more sensitive to previous methods, but it is limited by the difficulty to locate the origin of the degeneration. In these experiments, the origin was often unknown. In the 1970s, a more advanced tracing technique was developed and became the standard that is still in use today. Tract tracing also combines in vivo and post-mortem processing. But instead of introducing degeneration, it relies on the uptake of injected molecules by the neurons in the regions of interest. As shown in this figure, a neurotracer was injected into the inferior temporal lobe. The tracer molecule traveled inside the axons in the intact, unleashed brain. The animal was then sacrificed, and the brain was sliced and prepared for staining. Chemical reactions in the staining process made the trajectories of the tracer visible under the microscope. So we can trace its origin, passage, and termination. By going the stained brain slices one after the other under the microscope, one can reconstruct the full details of the fiber pathway being studied. Thus, tract tracing is a highly precise and highly sensitive method to study brain activity at a single axon level. There are 3 types of labeling in tract tracing that are most important for inferring brain connections. The first is the labeling of neuron bodies. Tracers can travel upstream from the injection site, or so-called retrogradely. The labeled neurons will tell us from which brain areas the injection site received input. The second type of labeling is the terminal fields. These are the destination of tracers that traveled downstream from the injection site, or so-called anterogradely. The terminal fields indicate to which brain areas the neurons from the injection site project to. Thus, these are the output destinations. The third type of labeling is fibers in the white matter. These sometimes show up as a very bright stick, but most often, these are fine lines in the deep white matter. By looking at these fine lines under the microscope and going from slide to slide, one can build the trajectory of fiber pathways in the white matter. These fibers tell us the relative position of the injection-specific connections within the major fiber bundles. Thus, a tract tracing experiment can provide very rich information regarding the connectivity of a particular injection. We can infer its input, its output, and the route of its connections. Now, coming back to the question about how our knowledge from animal studies can help understand the human brain. The information provided by tract tracing is based on direct measurement of the fiber pathways. It can be treated as the ground truth. And non-human primates are the closest relatives to humans among experimental animals. Thus, under the assumption of homology, we've already gained a lot of insights from decades of tract tracing studies. But the question is, can we do better than that? Is there a way to quantitatively and systematically translate the ground truth into humans? The answer is yes, and the promising approach is to use non-human primate imaging as a translational step. Recently, animal neuroimaging is growing through rapid development. It is now possible to acquire high-resolution, diffusion-weighted images, both in vivo and post-mortem, from animal brains. So, a potential strategy for translational neuroanatomy can follow 3 general steps. First, one can compare tract tracing and tractography results in the same species. In this case, we can look at both in non-human primates. We can rule out cross-species difference and focus on the discrepancies caused by methodological differences. We can then try to reproduce the tractor tracing results by adjust the modeling and tractography algorithms. Finally, with the adjusted algorithms, we can explore the homologous pathways in the human brain without being confounded by the methodological discrepancy. This strategy to translate anatomical ground truth into studying human brain connectivity has been successful in several studies. Now back to the problem we mentioned earlier about deep brain stimulation in OCD patients. We mentioned that it is important to accurately target the anterior limb of the internal capsule, or ALIC for short. The reason is that fibers connecting the brain regions that are relevant to OCD symptoms occupy subregions in the ALIC in a topographic manner, such that fibers originated from the ventral medial part of the brain will occupy the ventral portion of the ALIC. And the fibers from anterior cingulate cortex will occupy a more dorsal portion. And fibers from the dorsal prefrontal cortex will occupy an even more dorsal region in the ALIC. So in the paper by Safadi et al., 2018, as shown here, the authors first mapped out these topographical patterns in monkeys with tract tracing. And they compared monkey tractography and tract tracing data to rule out the false positive pathways. With the adjusted tractography results, they identified a highly consistent topography of the human ALIC, as shown in this figure. The red marks fibers from the ventral medial prefrontal cortex, yellow are fibers from the anterior cingulate, and green are fibers from the dorsal medial prefrontal cortex. These fiber patterns followed the same dorsal ventral pattern that was found with tract tracing in monkeys. Another example is a study on the hub region in the anterior cingulate. In this study, the authors first located a region in the rostral tip of the cingulate sulcus, where the injection resulted in an extensive connectivity pattern with a diverse range of frontal areas. The authors then replicated this convergent connectivity pattern with tractography, firstly in monkeys and then later in humans. As shown in these two figures, each red dot is an individual subject, and the results consistently show the most convergent connectivity pattern comes to the rostral tip of the cingulate sulcus, and that pattern is consistently located in the human anterior cingulate. So these studies suggest that cross-species and cross-modal integration can be achieved by using monkey diffusion tractography as an intermediate step. We are now in an exciting era where massive amounts of multimodal data are becoming publicly available. Many of the human imaging databases are already well known and well used. For example, Open Neural and the Human Connectome project. I especially want to point out some of the fast developing non-human primate data resources. The PrimeDE hosts monkey imaging data, including structural, functional, and diffusion for both task and resting states. It is now growing very fast. COCOMAC has been there for a while. It provides cytoarchitectonic and connectional information that covers an extensive range of brain areas. CoordNets is a tract tracing database focusing on the connection information. MacBrain resource is hosted at Yale. It contains a large collection of brain tissues as a legacy from the Goldman-Rakich lab. You can access these tissues based on request. On the other hand, big data brings big challenges. Analyzing tract tracing tissues is a very labor-intensive job. It requires examination under the microscope at a high resolution, which can take weeks to finish. One of the challenges that attracts computer scientists is to automate this process by applying machine learning algorithms to the microscopic images. However, it's less straightforward in practice than it sounds, particularly because the large data size. It is very common for a single image of a brain slice to exceed 5 gigabytes, for example. The automated tract tracing is an active research area right now. And there is, of course, an ongoing challenge from cross-species difference. We still haven't fully understood how exactly the monkey and human brains differ in their connectivity. Relatedly, when using tract tracing to correct for tractography algorithms, the problems are highly specific to the pathway being examined and have to be fixed case by case. Can we find a generalizable algorithm that is applicable to a more general range of pathways in both species? These are all open questions to be figured out in the future. Finally, I want to thank you for going through this anatomical intro at the DIPY Workshop. This is meant as a reminder that despite the complex computational problems tractography faces, the ultimate question is a biological one. That is precisely where a specific connection originates and terminates in the brain. And I hope you enjoy the rest of the workshop, in which you'll learn about the tools for tackling this question. And thank you.